There are several different ways that cells can repair their DNA, and we'll go through them in order. So proofreading and mismatch repair, post-replication repair in the SOS system, photoreactivation repair, base and nucleotide excision repair, and double-strand base repair in eukaryotes. So in proofreading and mismatch repair, this happens something that is just going to happen naturally. We have DNA polymerase will go along during DNA replication. It can remove incorrectly inserted nucleotides. It's going to recognize them, remove them. The correct one is put in by the DNA polymerase 3. If the proofreading fails, and it does fail somewhat, maybe 1 in 100,000 base pairs, mismatch repair becomes activated. Mismatches are detected, cut, removed. Certain enzymes called endonucleases and exonucleases. The correct nucleotide is then inserted by DNA polymerase. So it's literally a little cut and snip replaced with the correct base. There is a strong link between defective mismatch repair and cancer that has been documented. In post-replication repair and the SOS repair system, it's kind of like last resort. Post-replication repair is going to respond after the DNA damage has escaped the initial repair and has failed to be completely replicated. So what you can see here, there's a lesion. This may have happened due to UV irradiation causing a thymine dimer. The DNA is unwound. You're going to have replication in this direction, no problem. Here it's going to notice that there's a problem. It's going to skip over it, skip over that lesion. When it is trying to replicate it, there may be a new gap to form here. Okay, it can be filled in fairly easily by the DNA polymerase and DNA ligase. The SOS repair system is pretty much the last resort to minimize the DNA damage. So the DNA synthesis becomes error prone. It's going to start to insert random and incorrect nucleotides in places that would normally stall this DNA replication. The SOS repair itself can become mutagenic, but it can also allow cells to survive with DNA damage that otherwise might want to try to literally commit cell suicide. And we're going to talk more about that process a little bit later, called apoptosis. In photoreactivation repair, this is where we might see something like that thymine dimer again forming. In this case, it's going to involve a photoreactivation enzyme. It's going to cleave the bond between these thymine dimers. That enzyme absorbs a photon of light. Humans and other organisms, though, do lack this photoreactivation repair, but it is predominant in things like some prokaryotes. In base and nucleotide excision repair, the light-dependent DNA repair mechanisms that exist in the prokaryotes and eukaryotes are going to involve this excision repair. We have an exonuclease, which will recognize and cut that problem area. DNA polymerase, again, important enzyme, will insert those complementary nucleotides into the missing gap. DNA ligase is an important enzyme for sealing any gaps or nicks in that DNA. There are two types of this excision repair. You can excise just a single bases, and that would be base excision repair. And so again, just going to correct DNA containing a damaged, one single damaged DNA base. In nucleotide excision repair, we'll repair more bulky lesions that might alter or distort that double helix. So in a base excision repair, here you can see this is mismatched here with a uracil DNA. DNA glycosylase will recognize that this isn't supposed to be in DNA, will excise that incorrect base. There's going to be an enzyme that will recognize that lesion, will nick that strand. It will then, DNA polymerase can then, and DNA ligase can fix that gap. And at this point, that's mismatch has been repaired. In nucleotide excision repair is usually going to be for a little bit larger lesions where the DNA is damaged. The nuclease will excise a lesion and also some regions upstream and downstream of that. The DNA polymerase is going to fill that gap. The gap can be sealed with DNA ligase, and this normal pairing then should be restored. So another case study, xeroderma pigmentosum is a rare genetic disorder, and it arises though due to defects in these nucleotide excision repair pathways, and by mutations in more than one gene. Individuals who have this disorder, thus, there may accumulate mutations at a higher than average rate because their body cannot fix those problems. So these affected individuals are going to exhibit severe skin abnormalities, skin cancers, wide range of other symptoms. They have a 2,000-fold higher rate of cancer than somebody without this disorder. And again, it's just because their body is going to be able to accumulate these additional mutations and not be able to fix the problem through this particular mechanism. In double-strand base pair repair in eukaryotes, what can happen is exposure to ionizing radiation can lead to both strands of DNA being cleaved and broken. And this results in chromosomal rearrangements, cancers, cell death. What happens is you're going to reattach these two broken DNA strands through homologous recombination and repair, through non-homologous end joining. And these just occur during the interphase of the cell cycle. So late S phase, early G2 phase, or G1 phase. And what you can see here, a double-stranded DNA break. The break is going to be detected. The 5' ends are going to be digested. So this break happened here, but they're going to digest a little bit of overlap here, a little bit of the overlap here. The 3' end is going to go on to the homologous region of the sister chromatid. Okay, so here's the sister chromatids. It's going to use that as a template. This DNA synthesis is going to come across that damaged region. It's going to go across here. And now you have this problem fixed. The gaps are going to be filled, and they're going to be ligated. And you can see in purple here is going to be where the new nucleotides have been laid down. And this right here was where the initial break was. And you can see that homologous chromosome hasn't really been affected at all, or the sister chromatids haven't really been affected at all.